Okay, so imagine, like, you're a detective, right? But instead of like a crime scene, you're investigating the world of cells, like hunting for clues, but inside individual cells. That's kind of the power of flow cytometry. And today we're going to take a deep dive into flow cytometry gating,
Mhm.
the technique that helps us solve those tiny mysteries.
It's like we had this high-tech sorting hat, but for cells. It lets us separate them and analyze them based on like their own special features.
We're using this awesome guide from Assay Genie. They're a biotech company, and it's really helping us understand this complex world. Think of this deep dive as like your detective's handbook for flow cytometry gating. Okay, so you've just finished a flow cytometry experiment. You're looking at tons of data, and you need to find the important stuff, the signal within all the noise. That's where gating comes in, right?
Yeah, so gating lets us set up filters,
Yeah.
kind of like, custom-made sieves, to isolate specific cells from like all that data. We're sorting cells based on their size, their internal structure, and even if they have certain proteins.
Okay, so where do we even begin with gating? It's like looking at a galaxy of data points.
It all starts with like getting a basic ID for each cell. And we
Yeah.
do that by looking at forward scatter and side scatter.
FSC and SSC.
Yeah. These parameters, they basically give us a sneak peek into the cell's physical like characteristics.
So it's like a mugshot telling us, is it a big cell? Is it a small cell?
Exactly. So FSC that measures cell size. The bigger the cell, the more light it scatters forward, and that gives us a higher FSC value. SSC, though, that tells us about the cell's complexity on the inside, its granularity.
Oh, so a cell that's like packed with organelles would have a high SSC.
Exactly. Now, when we plot FSC against SSC, what we end up with is like a map, a visual map of our entire cell population. And from this map, we can start drawing gates, basically boundaries, to separate, the different groups of cells. It's like sectioning off different parts of our city based on what kind of cells live there.
Okay, I'm picturing a scatter plot. So each dot's a cell, and we're drawing fences around specific neighborhoods. But what about those doublets? I've heard those can really mess things up.
Yeah, you got to watch out for those doublets. They're like two cells stuck together pretending to be one big cell. And that can lead to some pretty bad data.
So if we're like counting cells or studying specific cell populations, doublets can really throw things off. It's like counting two criminals disguised as one. How do we avoid that?
Well, we've got ways to expose them. We use a combination of FSC parameters, FSCA, FSCH, and FSCW.
Oh, so we're looking at the cell size from different angles?
Exactly. Single cells have a predictable relationship between these FSC values, but doublets, because they're two cells smooshed together, they throw off those ratios. So by plotting those parameters against each other, we can spot the doublets.
Ah, so we can then draw a gate to exclude those doublets, making sure we're only analyzing data from single cells.
Yeah. But even after we get rid of the debris in the doublets, there's still another problem we need to address: dead cells.
Dead cells. What's the issue with those? I figured they'd just be floating around.
They can really mess with our data.
Mm.
They bind to antibodies non-specifically, and that can lead to like false positives.
So it's like finding a fingerprint, but it turns out to belong to someone who was never even there.
Exactly. So to get rid of these dead cells, we use something called viability dyes.
Viability dies. Sounds intense. How do those work?
They act like zombie detectors. They
Yeah.
can only get into cells with damaged membranes, so they stain only the dead cells. Makes them
Mhm.
stand out.
Okay, so we've marked our cellular zombies, but how do we actually see that in the data?
We plot the viability dye fluorescence against FSCA, and that creates a visual separation between the living cells and, those that have passed on. We can draw a gate to exclude the dead cells, leaving us with a clean data set of only the living cells.
It's like making sure we're only looking at clues left by living suspects. So we've cleared out the debris, the doublets, the dead cells. Now we're left with just viable single cells. Feels like we've narrowed down our suspect list. What's the next step?
Now we get to identify the specific cell types. And for that, we need antibodies.
Antibodies, those are the tiny protein detectives that, recognize and bind to specific targets, right?
Exactly. And we attach a fluorescent tag to each antibody,
Yeah.
so they light up when they find their target. This lets us identify and quantify different cell populations within our sample based on what proteins they have.
So it's like giving each antibody a flashlight to find its target. If we're looking at T cells, for example, we'd use antibodies that bind to CD3, which is on all T cells.
Right. And then we can analyze the fluorescence signal And that tells us the percentage of CD3 positive cells in the sample.
And if we want to look deeper into the T cells, we could use antibodies against like CD4 and CD8 to tell the difference between helper T cells and cytotoxic T cells.
Exactly. It's like using different badges to identify the different players in our, in our cellular world.
So we're not just looking at a jumble of cells anymore. We're starting to identify specific groups and what they do.
Exactly. And that's just the beginning. We can use a bunch of antibodies all at the same time to create these detailed profiles of different cell types, and it lets us uncover these really intricate relationships within our, cellular world. But even with the best detectives, mistakes happen. So we rely on controls to make sure our investigation is accurate. And one of the most important controls we have is the FMO control.
FMO control. What's that?
It stands for fluorescence -1. And it's super important for dealing with something called fluorescent spillover.
Fluorescent spillover. What's that like when one fluorescent signal like messes with another?
Yeah. Imagine you're trying to listen to the radio, but there's another station on a close frequency, and it's bleeding through, making it hard to hear clearly. That's basically what fluorescent spillover does.
Oh, I see. So it's like two witnesses talking at the same time, and it's hard to understand what each one is saying.
Exactly. Let's say you're using two different antibodies, one tagged with a green fluorescent dye, the other with a red dye. If there's spillover between the green and red channels, it might look like some cells are expressing both proteins, but actually they're only expressing one.
Ah, so it's like a false confession because of the overlapping signals. How do FMO controls help with that?
They're like setting up a control experiment for each color. We take a sample that has all the antibodies except for one. So like we have a green, red, and blue panel. We'd have 3 FMO controls, one missing each color.
So we're testing each color on its own to see how much it like spills into the other channels.
Exactly. And this tells us how much signal bleeding is happening. And that lets us correct for it, make sure we're interpreting the data correctly.
So FMO controls help us separate the true signal from all the background noise.
Exactly. They're super important for, for getting accurate data, especially in these complex experiments where we're using multiple colors, and spillover can be a big issue.
Yeah.
So welcome back to our deep dive into flow cytometry gating.
It's pretty amazing, how much we can learn about these individual cells just by like carefully looking at these scatter plots and, fluorescent signals and all that.
It's like you have this superpower. You can like zoom in to this hidden world and see how individual cells are, behaving, how they interact, all that.
Exactly. And it's not just, pretty pictures and complex graphs. The information we get from flow cytometry gating actually has like real world uses.
Absolutely. Think about it. Understanding, how the immune system responds, developing drugs, figuring out if someone has a disease, is the treatment working, all that. Flow cytometry is like at the center of it all, both in research and in, actual medicine.
Okay, so let's continue exploring this powerful technique. Last time we talked about the basics. Getting rid of debris, dealing with those doublets, and using antibodies to find specific cells. But I'm guessing there's more to gating than just plotting a few parameters.
Oh, yeah, definitely. Gating isn't like a one-size-fits-all thing. We have a bunch of different strategies and techniques to choose from, and it really depends on what we're trying to find out.
Yeah.
And how complex the situation is.
So it's like picking the right detective for the case. Sometimes you need, like, a specialist.
Exactly. And one of those special approaches is sequential gating. It's kind of like peeling back the layers of an onion. We can see hidden stuff within our cells.
Oh, I like that. So we start with, a broad gate to capture all the suspects, and then we slowly narrow it down using more markers.
Exactly.
Yeah.
Like, let's go back to the T cells. We might start by gating on, all lymphocytes based on their FSC and SSC.
Okay, so we've got all our lymphocytes. Now what?
Then we use a CD3 antibody, so we can focus on the T cells within that group.
And then we could use the CD4 and CD8 antibodies to tell the difference between the helper T cells and the cytotoxic T cells, right?
Exactly. We're figuring out who the leaders are and who the, followers are. And within those groups, we can even look for other markers like activation markers, things like that, to understand how those T cells are acting, what they might be doing.
So it's like analyzing their fingerprints, looking for weapons, any evidence that can help us understand what they're up to.
Exactly. Sequential gating helps us break down this complex network of cells step by step, and then we can see how the different players are related.
It's like we're creating a family tree for ourselves.
Now imagine you've spent hours, carefully putting together this case, building your theory. Don't you think you'd want to double check it? Make sure you didn't miss anything?
Oh, for sure. Like, one tiny mistake and the whole thing could fall apart.
Right. Well, in flow cytometry, we can double check using something called backgating.
Backgating? Is that like time travel?
Not quite, but it does let us look back
Mm.
and check that our strategy is right. It's like retracing our steps, making sure we didn't leave anyone out or put someone in the wrong group.
Oh, okay. So it's like, reviewing the security footage, see if anyone slipped through.
Exactly. Let's say we've gated on a certain group of cells based on, one particular marker. Backgating lets us see where those cells are on, like, other plots.
So we're comparing our findings, making sure everything matches up.
Yeah. Let's say we've gated on CD4 plus T cells. We think we've got all our helper T cells right.
Those are the cells that like organize the immune response.
Right. But then we use backgating, and we see that a lot of those CD4 plus cells, they're also positive for CD8.
Wait, but CD4 and CD8 are supposed to be on different T cells. It's like our suspect has two identities.
Exactly. And that tells us that our initial CD4 gate might not be as accurate as we thought. Maybe there's a group of T cells that have both markers, or maybe there's some spillover.
So backgating helps us catch those inconsistencies where maybe our initial idea was wrong.
Exactly. It's really helpful for making sure our investigation is, accurate and reliable.
It's like we're building a whole forensic lab for ourselves.
And speaking of forensics, remember we talked about fluorescent spillover before?
Yeah.
Well, we have a really important process for dealing with that. Compensation.
Right, compensation. That's where we adjust for like the overlapping signals from the dyes.
Yeah, like you're adjusting the volume on different instruments to hear each one clearly.
Exactly. So fluorescent spillover, that can create like false positives, right? Making it seem like a cell has a bunch of proteins when it really only has one.
Right. It's like those shadows that can like distort things.
So compensation is how we adjust for that spillover so we can really see like the true fluorescence.
Exactly. It's like calibrating our instruments, fine-tuning them. Now, compensation might sound complicated, but most flow cytometers have software that does it for us.
Phew. That's good to hear. I thought we'd need like a physics degree.
No, it's not that bad. Basically, we run single stain controls for each color.
Single stained controls. What are those?
So it's samples of cells that have been stained with only one of the dyes we're using. So like if we have green, red, and blue, we'll have 3 controls, one with just green, one with just red, one with just blue.
Okay, so it's like listening to each instrument in an orchestra on its own.
Exactly. We run these controls, and the software analyzes them. Then it uses that information to calculate compensation values. Those are basically correction factors, and they account for the spillover between the different dyes.
Oh, okay, so it's like a sound engineer adjusting the levels so everything's balanced and we can hear each instrument clearly.
That's a great way to put it. And then once we have those compensation values, we use them on our actual experimental data to correct for the spillover, and that gives us like a really clear and accurate picture.
So compensation, it's like super important then, right? Especially when you're doing these complicated experiments with lots of colors.
Absolutely. It's all about making sure our data is good and that we're, drawing the right conclusions from our cellular investigation.
And we're back for the last part of our deep dive into flow cytometry gating. It's been like a wild ride, We started feeling kind of lost in all that data, but now we can find specific cells, and we've even learned some cool cellular secrets.
Yeah, it's really amazing what we can do when we have, the right tools and know the right techniques.
For sure. And speaking of tools, I've heard about this thing called dimensionality reduction. Sounds kind of sci-fi, but what is it exactly?
That does sound a bit futuristic, right? Basically, dimensionality reduction helps us like simplify these really complex data sets and lets us see high-dimensional data.
Okay, I'm all about keeping things simple, especially with data. But what exactly does like high-dimensional data even mean when we're talking about flow cytometry?
So remember how we measure, a bunch of different things for each cell, like the size, granularity, and all the different proteins they have?
That's like giving each cell like a detailed profile.
Exactly. And each of those measurements adds another dimension to our data. So if we're measuring 10 things, we're working with 10-dimensional data.
10 dimensions. Whoa, that's a lot. I can barely imagine 3 dimensions.
It's definitely hard for us to picture, right? And that's where dimensionality reduction comes in. It takes all those dimensions and it kind of squishes them down into something we can actually see, like 2 or 3 dimensions.
Oh, so it's like making a simple map of, a really complex area We might lose some details, but we get a better overall view.
Yeah, that's a good way to think about it. There's, a bunch of different dimensionality reduction methods, but some of the most common ones are PCA, that stands for Principal Component Analysis, and TSNE, which is T-distributed stochastic neighbor embedding.
Those are some long names. So what do they actually do?
They basically help us find patterns and groups in our data that, we might not see if we're looking at 10 dimensions at once.
So instead of looking at each measurement on its own, we can see how they all work together and group cells that are similar.
Exactly. And that can be really helpful for finding rare cells or even, new types of cells that we might have missed with normal gating.
So it's like using a special lens to see hidden stuff in our data.
Yeah. And the best part is once we've used, dimensionality reduction to find these clusters, we can go back to our original data and gate on them.
Oh, so we're using dimensionality reduction to scout things out and then zooming in for a closer look.
Exactly. It's a really powerful way to, improve our gating and maybe even discover some new things.
This is really cool. It's like we're giving our detective kit some serious high-tech upgrades.
And speaking of upgrades, let's talk about another advanced technique, Boolean gating.
Boolean gating. That sounds kind of like computer programming.
It does have a bit of a programming vibe, yeah. It lets us define cell populations based on combinations of markers, and we use these things called logical operators, like A and D, or R, and not T.
Oh, okay, now I see the programming connection. Can you give us an example?
Sure. Let's say we're interested in regulatory T cells. Those are called TREGs. They're super important for keeping the immune system in check.
Right. They make sure things don't get out of control.
Exactly. And TREGs, they have a specific combination of markers, including CD4 and CD25. With Boolean gating, we can make a gate that only includes cells that are positive for both of those markers at the same time. So our condition would be CD4, A and D, CD25.
Oh, so only cells that have both get in.
Exactly. We can also use OR gates to be a bit more flexible. Like if we wanted to look at all the lymphocytes, which includes both T cells and B cells, we could use an OR gate. So our condition would be CD3, which is the T cell marker, or our CD19, the B cell marker.
So that would include any cell with either one of those markers.
Exactly. And then there's NOT gates, which let us, exclude certain cells. Like if we're studying live cells, we can use a NOT gate to get rid of all those dead cells that we tagged with a viability die.
Oh, so it's like no dead cells allowed in here.
Yeah. Boolean gating gives us, a lot of control over how we define our cell populations.
It's like having these filters so we can search for exactly what we want in our cellular database.
Right. And it's really useful when we're looking for, rare cells or trying to understand complex populations.
This is all so powerful, but it sounds like it takes a lot of planning, right? And a deep understanding of all these markers and stuff.
Yeah, definitely. When you design a gating strategy, it's like coming up with, a good plan. You got to think about your research question, the markers, and, the biology of what you're studying.
So it's like being a detective, but also a strategist.
Exactly. So, do you want to go over some practical tips for our listeners? Things they should remember for effective gating.
Yeah, definitely. It's always good to review the notes before we head out into the field.
Okay, first, always start with a clear idea of what you're trying to find. That'll help you choose the right markers and, guide your whole strategy.
Right. You need a clear goal, otherwise you can get lost.
And remember how important controls are. We talked about FMO controls for compensation, but there's other types too. Unstained controls tell us about the background fluorescence, and isotype controls help us see if antibodies are binding non-specifically.
So those help us make sure that what we're seeing is real.
Exactly. And whenever you can, use sequential gating. Start with broad gates and then refine it step by step. It's a really organized way to find your target cells.
It's like using sieves to get rid of all the junk and keep the good stuff.
Exactly. And lastly, don't be afraid to try different gating strategies. What works for one experiment might not be the best for another one. It's all about finding what gives you the clearest picture.
So it's a process of, trying things out, making adjustments, getting better and better.
Exactly. The more you do it, the easier it gets.
This deep dive into flow cytometry gating has been a real adventure. We've gone over the basics, learned some advanced techniques, and, figured out how to navigate all that complex data.
It's been great exploring this with you.
And to all our listeners out there, remember that flow cytometry isn't just about, abstract numbers. It's about understanding diseases, developing new treatments, and making people healthier.
It's a field with so much potential for, discoveries and innovation.
Absolutely. So as we wrap up this deep dive, I want you to think about what questions you have. What
Yeah.
mysteries about cells do you want to solve? What hidden patterns are waiting to be found? The tools are out there. You just need to, start exploring.